Hi, I'm Kemp. I'm from the University of Bristol, and I'm a scientific researcher looking at the neuroprotective mechanisms of bone marrow stem cells for friedreich's ataxia. My poster is looking at a particular mechanism of how bone marrow cells can be used as a treatment for friedreich's ataxia, and we're looking at how bone marrow cells can fuse with neurons in the brain as a potential gene therapy for the disease. Here you can see markers of cell fusion within the adult brain, so you can see these cells have two nuclei, which is an indicator of cell fusion. And we wanted to look at the therapeutic properties of these cells in a mouse model of friedreich's ataxia, particularly the YG8R model. So our work looks at giving these Friedreich's mice a bone marrow transplant. These are transplant of a bone marrow cells that express a green fluorescent protein, so to be used as a marker so we can see them throughout the nervous system. Here we can see some pictures of the dorsal ganglia, the spinal cord, and also the cerebellum, seeing that there's a lot of these green cells integrating within the nervous system. Looking a bit more in detail, so we can look in the dorsal ganglia, we see that they express markers for satellite cells. We also see that these green cells here, they can actually wrap around axons, so they look like myelinating swan cells, so they can possibly increase, help with nerve conduction. Looking a bit more at cell fusion itself, we can see that these cells fuse with neurons within the dorsal ganglia. You can see quite nicely that there's a nice green fluorescent Purkinje cell within the cerebellum. And we also see various other cells, so cells within the dentate nucleus. So what we did is we give these mice a sex mismatched transplant. So what we do is we transplant male cells into female recipients, and using the Y chromosome we can look at how the nucleus can integrate within neurons. So here's some pictures down here. So we can use a method called fluorescent in situ hybridization. So we can look at tagging the X and Y chromosomes, and here we can see that we've tagged the Y chromosome using a fluorescent marker, and we can see that the Y chromosome is found within the nucleus of things like Purkinje cells and also dorsal ganglion neurons, so showing that genetic material has been inserted within the host neurons within the mouse. So this gives an indication that we might be able to use bone marrow stem cell fusion as a method of inserting genes, particularly corrected frataxin genes, as a mode of gene therapy for the disease. So with these interesting results, we've already had successful funding from both Ataxia UK and FARA. So they've co-funded a project looking at how we can use bone marrow stem cell mobilizing drugs in patients with friedreich's ataxia. This work is starting as a pilot phase 1 trial, looking at the safety and efficacy of these mobilizing drugs in patients with friedreich's ataxia. So we should be starting this within the next few months. These patients will be given an infusion of GCSF. This is the stem cell mobilizing drug that's commonly used for peripheral blood stem cell harvests in patients. And then we'll be looking at various biomarkers. So we'll be looking to see if it increases things like frataxin, PGC1 alpha, and various other frataxin associated molecules. With this, hopefully, if we see some results that what we see replicate within the mice, and hopefully we can take this onto a phase 2 trial, and hopefully we can see some really exciting results from that.